So our lab studies interactions between megakaryocytes and bone cells and the effect that has on bone phenotype. One important thing our lab's actually shown, and other labs as well, is that with a high number of megakaryocytes you get a high bone mass phenotype. And it's done because megakaryocytes interact with osteoblasts as well as osteoclasts. So they actually activate the osteoblasts and then inhibit the osteoclasts. And in certain mouse models you get a really high bone mass phenotype because you're doing this. So that was really interesting. What we found is that mice that are overexpressing thyromopoietin, for example, have a high number of megakaryocytes and then a high bone mass phenotype. So one project that I've been working on is looking at CMPL, which is the receptor for thyromopoietin, and how a knockout of this would affect the bone phenotype. So what we would expect in a mouse that has no CMPL, since CMPL is necessary for thyromopoietin to bind to megakaryocytes and get a large number of megakaryocytes, you would think there would be a low number of megakaryocytes in these mice. And we do see that, but what we would think is with a low number of megakaryocytes that we would have a loss of that interaction between megakaryocytes and osteoblasts, and therefore a low bone phenotype, so low bone mass. But we actually found in this mouse that doesn't have that CMPL on the surface of the cells is that they have a normal or high bone mass phenotype. So that was interesting to us, and that really set the foundation for trying to understand what's happening in this mouse model. So what we actually found is that CMPL, which was previously thought to be highly restricted to hematopoietic cells, megakaryocytes specifically, was also expressed on osteoblast progenitors, osteoblasts, and osteoclast progenitors. So that kind of opened the door to explore what's the role of CMPL expression on these bone cells, and is it possible that it's having some sort of other regulation. So this has been a project that's been going on for quite a few years. I haven't been the only one that's been working on it, but I've really been able to see this project through for a couple of years, and that's been really rewarding to be able to do something like that, kind of seeing this story play out over the years. So we've done a lot of examination on osteoblasts and osteoclasts and how CMPL is regulating both of these, and done some really interesting experiments where we do co-cultures where we can culture osteoblasts in the presence of bone marrow macrophages, and we can see how the osteoblast presence and the genotype of the osteoblast is affecting osteoclast formation, which is kind of simulating what happens in the bone microenvironment. You have a lot of complex interactions, there's a ton of different cells there, so we can simplify it down but still get some of the interactions that happen in bone, and we can do that in vitro.